but only about 10%. Nutrition, if you knew the worksheet on nutrition, the things that I highlighted on nutrition, would be okay. Electrolyte fluid balance, there's probably a couple questions on that. So I'm going to tackle today the elephant in the room. The elephant in the room is the cardiovascular system. So, unfortunately, I'm hesitant to say this, but there are still some folks in here that on the last exam still did not know the blood flow through the heart. So it will be the first question on the final exam. So we've gotten to the point now where we've gone through 4 lecture exams, and now a final. So we're going to learn today the blood flow through the heart. For all those of you who continually do not get it. What?
It's probably not any of these people. It's probably somebody who's not here that keeps getting it wrong.
You know what? You're probably right, but for the benefit of, as people say, for the benefit of Mr. Kite, I am going to go through this for those of you who can maybe pass the information along to your colleagues, right? Who somehow have a mental block whereby they cannot figure out the way the question is worded or something to that effect. They can't put 2 and 2 together correctly. I leave a couple of blanks. Obviously, on those type of questions, the key word is the last word, right? Where the blood is going to ultimately be shunted to the lung or to the body or whatever. The last word in the sentence is really the key word, right? Because everything before that determines where the blood is going to end up. So, just from the beginning, way back in, when did we start? January?
Yeah.
Way back in January, we started very carefully understanding blood flow. And I said it's really one of the most important things you're going to have to know. If you know nothing else, I can't reduce it to that, but it's very important. It's very significant that you know normal blood flow. So we know that there are going to be basically 2 circuits involved. There's going to be a systemic circuit and there's going to be a pulmonary circuit. And in the pulmonary circuit, vessels which normally carry oxygenated blood do not. And vessels which obviously carry deoxygenated blood do not. So they're reversed. We have learned from day 1 when you take the first biology course, the arteries carry oxygenated blood. And we have learned from day 1 that arteries take blood away from the heart. Artery away. A. We've also learned that veins carry deoxygenated blood, and veins take blood toward the heart. So if we keep those concepts in our mind, the away part and toward part, we can figure out the circuits. The important thing to remember is that the heart does not have any brain. The heart is just a pump. It's a mechanical pump. It's a very, very important pump, but nevertheless it's a pump. And the pump of the human heart in the adult has 2 sides. It has a left and a right. Both are individual pumps. One side pumps deoxygenated blood, and one side pumps oxygenated blood. In order for the heart to work as a pump, we have to somehow get blood back to it in order for it to work. So how do we do that? Well, we know that there's a whole pile of veins scattered throughout our entire body called systemic veins. And these veins are now going to be taking blood back to the heart, like normal veins. And what is going on now is the fact that we have big vessels, namely the inferior vena cava, the superior vena cava, and those blood vessels are taking blood which has now been utilized by tissues. Down here somewhere in the capillary system of our body, we have oxygen being delivered to tissues. And tissues utilize the oxygen and they make metabolic waste. They take the oxygen from the blood, from the hemoglobin. It's called unloading of hemoglobin, or unloading of oxygen. And when the oxygen is unloaded from the hemoglobin molecule to the tissues, they produce carbon dioxide, and they produce metabolites. Somehow that blood now is void of its oxygen content. So we call it deoxygenated blood. So the deoxygenated blood now has to return somehow to the heart. So how do we get it there? We get it there by major trunks of our venous circuit. So we have the inferior vena cava and the superior vena cava bringing down blood to the right side of the heart. So we have to know that blood is returned to the right side, and basically we have to know that it goes through various chambers. The two chambers that lie superior in the heart are the atria. So the first part of that blood has to go into the right atrium. It has to. And we also know that the heart is itself a muscle and utilizes oxygen. So we know that oxygen now has to be utilized by cardiac cells, which is tissue. And then that in turn says that some of the oxygen is taken out of that bloodstream, and then that has to get put to the right atrium as well. So we know that there's another opening in the heart called the coronary sinus. So the three major points where deoxygenated blood is returned to the right side of the heart: superior vena cava, inferior vena cava, and coronary sinus. Now we have all the deoxygenated blood in one spot. From the right atrium, it has to be delivered to the right ventricle, which lies inferior to it. And we're going to learn about, further on in a little bit of time here, we're going to learn about it has to go through a valve. We're going to talk a lot about that later. Just want to get blood flow. So now the blood has to go from right atrium to right ventricle. Now the right ventricle is the pump. The atria are receiving chambers. The ventricles are pumping chambers. So now the ventricle is going to be the pump. And that is going to take deoxygenated blood. No miracle has happened. It hasn't been oxygenated yet. So it's going to take the deoxygenated blood and it's going to pump it through, now it's going to pump it through an artery because it's taking blood away from the heart. This is the point where, in the adult, deoxygenated blood is carried in arteries. We are now entering the pulmonary circuit. So the pulmonary trunk, or pulmonary artery, takes the deoxygenated blood and delivers it to the lung. And we now know in birth, our adult form, we have functional lungs. And we've learned that the lung is the place where oxygenation takes place. So now we have the deoxygenated blood in the lung, and the lung is doing its job. The The lung is taking over now and oxygenating that blood. Now the blood has to be returned again. Now we have oxygenated blood coming back to the left side of the heart. And we have it being returned now by veins, because now we know veins carry blood toward the heart. So our lungs are lateral to our heart, and now we have to have vessels bringing the blood back toward the heart, namely veins. This is the case, one of only two cases in the human adult body where veins carry oxygenated blood. The other vein I'm going to talk about on Monday. So this is one of the only two places where veins carry oxygenated blood, and now the oxygenated blood is put back to the left side of the heart. From the left side of the heart, again, it has to enter the superior chamber, which is the left atria. And then it gets delivered to the ventricle, the left ventricle, which is the pump on the left side. And the pump now pushes the blood through another valve into the aorta. The aorta is the largest blood vessel we have in our human body. The aorta now is carrying oxygenated blood to the entire rest of our body. It's taking blood now away from the heart, so the aorta has to be an artery, and it takes blood into smaller and smaller blood vessels, still arteries, bringing blood to the various capillary networks in our body, where there's tissue exchange taking place. Oxygen going to the tissues, carbon dioxide leaving the tissues, getting put into the venous circuit. Any question on that? No? So I can assume that everyone here now today is going to get the first question on the final exam 100% correct.
Yes.
Yes?
Yes.
It's a big assumption. It's a big assumption. But we have to know the blood flow through the heart. The blood flow through the human heart in the adult form. You really have to know that. But it will be the first question on the final. I keep giving this question over and over. I change it slightly from here to there, hither and dither, but it's still the same deal, the same concept. Okay? We have to know the blood flow through the heart, right? I'm going to take your word for it. That can be foolhardy. All right. I'm going to go through a series of slides, and I'm going to make sure that I sort of follow along with your study guide. It says location, structure, valves. We talked a lot about valves. We talked a lot about the EKG. We talked a lot about hemodynamics. So I'm going to go through a few of the highlights that I think are important that you might see somewhere down the line, maybe on May 8th. We know the heart is in a very, very special and specific place in our human body. The specific place is called the middle mediastinum. It's not in the lateral mediastinum, it's not in the superior mediastinum, it's in the middle mediastinum. It is surrounded by a sac called the pericardium. It would be nice if you knew, for example, the layers of the pericardial sac. We have an outer fibrous layer, and then reflecting that back on the inside surface of that is a parietal pericardium. And then on the heart muscle itself, you have serous membrane that cannot be stripped off that. That's called the epicardium, or the visceral pericardium. So we know the heart has a nice sac surrounding it. Okay? It would be nice to know the pericardial sac. It would be nice to know where the heart resides in the human body, the middle mediastinum. The layers of the pericardial sac are very important. Those of you who looked at the cadaver, very, very clear on a human cadaver. The fibrous layer is the outermost layer. It is tough. You can't pull it apart. Right? I have to cut it with a pair of scissors or a scalpel. That creates a lot of clinical issues that we talked about, right? If we have an important concept of how the heart works, the heart is a pump. Pumps have to beat. They have to move. Heart's no different. So we have to know that between a couple of those layers, we have a nice space between the epicardium, which lies right on the heart muscle, and the parietal pericardial underside of the fibrous layer. And that's called the pericardial cavity. That cavity is filled with fluid. A little bit of fluid, but fluid. Right? And who wants to tell me what the fluid does?
Causes so that there's not friction?
It causes reduction in friction. So when the heart beats and moves, we don't have two layers of membrane coming together and sticking together and causing heat, causing friction. So the pericardial cavity is filled with a small amount of serous fluid. Now I told you before, we can have a problem with that. We can have too much fluid, or we can have too little fluid. Right? So there's conditions that have to be met. Not to get too clinical for the final exam, but we have to know that there's a pericardial cavity and it's supplied by the appropriate amount of serous fluid. The heart muscle itself is an interesting sort because the heart muscle is not just linear. The heart itself is a very, very relatively small structure, but it has to do a lot of work. And so the heart muscle is contrived in the heart so that it sort of acts like a dish towel, a wringing motion to squeeze every single drop of blood out that it possibly can. Right? So the heart muscle is an important, the architecture of the heart muscle is important to know. It would be good to know the first two branches off the ascending aorta. What are the first two branches off the ascending aorta? Remember now, the aorta comes directly from the left ventricle. So we have an ascending aorta, and then we have an arch of an aorta, and then we have a descending aorta, which lies posterior to the heart. What are the first two branches off the ascending aorta?
The coronary arteries.
Left and right coronary artery. It would be nice to know that. It's fundamentally important because the heart has to have a continual supply of oxygenated blood. So basically, it serves itself first. Right? I tell you it doesn't have a brain, but it's no dummy. Right? It takes the oxygenated blood right away before it passes any more into the aorta. So the first two branches off the ascending aorta go back directly to the heart. We know also that the heart, the left and right coronary artery divide into various branches. The most important one is this one right here, which lies in a very, very nice groove called the anterior interventricular groove, and that's referred to as what? Anterior interventricular artery, or LAD. Left anterior descending. That is very, very important. Probably one of the most important arteries. And it is a branch of a coronary artery. Okay? When you look at a human heart, you know that that groove separates right from left ventricle. So when you look at normally a human heart, the most structure you see on the anterior surface is the right ventricle. The veins are interesting, but we know that there's a whole pile of veins. We talked a lot about it in class where I've mentioned 3 big ones: the great cardiac vein, the middle vein, and the lesser vein. But they all join together. They all join together on the posterior surface of the heart, and they form an enlarged area, an enlarged vessel. And this enlarged vessel is going to be very, very important because that's the coronary sinus that enters the right atrium. So all the veins, and there's a lot of them, there's a lot of cardiac veins. I just mentioned 3 in class, but there's a lot of them on your heart. But they're all going to deliver blood to the coronary sinus, which is then returned to the right atrium. And the cycle starts of oxygenating it again. Right? We talked a lot about valves. So there will be some questions on valves. We spent a lot of time on valves. Valves are extremely important. On the right, we have the valve with 3 leaflets which separates atria from ventricle, and that's called the tricuspid valve. On the left, we have another valve with only 2 cusps, which separates left atrium from left ventricle, and that's called the bicuspid valve, or the mitral valve. Whether we're talking about left or right, we know that valves are important because we have valve leaflets attached to small threads called chordae tendineae. And those fine thread-like attachments are attached and anchored to other muscle in the heart called papillary muscle. So here's the question. Some of you got it wrong on the exam. I put this question on the exam. I probably will put it on the final. What is the function of the papillary muscle and chordae tendineae? Does it have anything to do with contracting the ventricle?
No.
Does it have anything to do with, say, changing the dynamic of how much blood gets into a ventricle or whatever? No. What is the major function of the chordae tendineae and the papillary muscle? The major function is that it puts tension on the valve leaflets. So when the pressure builds in the ventricles, the pumps, the pressure is building, the valve leaflets close and come together, and they have to be held in that position. The function of doing that is the chordae tendineae and papillary muscle. That is their function in life. If this is not accomplished correctly, the pressure in the ventricle is so high that it blows the valve leaflets open. And now we have blood going backward. We have blood pushed back into the atria, which we never want. What's that called? Regurgitation. So we never want that. So we talked a lot about valves. We talked a lot about how there's two ways to look at a valve. If it does not close properly and not held tightly and opens, we have an incompetent valve. If the valve does not open correctly to let blood flow from atria to ventricle, then we have a stenotic valve. So I would suggest you look and make sure you understand the mechanisms of valves. Now, the other important valves that we have to look at, which are dynamically completely different than the atrioventricular valves, the AV valves, are the semilunar valves. So we have valves in the pulmonary trunk and in the aorta. And these are semilunar. These are like little cups that catch the blood and hold the blood in place. So it doesn't flow back into the ventricle after ejection. So make sure you take a look at the valves. Valves are going to be on the exam. The mechanism of action between the AV and the semilunar is completely different. Remember we talked about it in class where I said the AV valves are more active, they're more mechanical because the papillary muscle has to contract, the chordae tendinii have to tighten and hold the valve in place. Whereas the semilunar valves are passive. When the blood starts flowing backward, they just catch the blood. They just open like little upside down umbrellas. So the mechanism of action is completely different between the valves. Here again is blood flow through the heart. We know that there's a big difference between the left ventricle and the right ventricle. The left ventricle has a lot more muscle to it. And you did that in your lab, right? Remember you looked at the left side of the heart and the right side of the heart. The left side of the heart has a lot of myocardium. Because it has to push blood everywhere. The right side of the heart, the ventricle on the right, only has to push blood to the lung, which is right next door. So the muscle is accommodating the amount of force needed to push the blood. What makes the heart important as a functional pump, this is going to be on the exam, somewhere along the exam, is that we know that the cardiac muscle is specialized muscle. And what makes it specialized is the fact that it has these heavy dark lines, which are called intercalated discs. So I would recommend that you look at what an intercalated disc is, make sure you understand what it does, how it's attached, and desmosomes, how they are little clamps that clamp the myocytes together. All of that makes the heart efficient and act as a functional syncytium. You should know that word, functional syncytium. Acts as a unit, it contracts as a unit. When the heart contracts, we want all the muscles to work, all the muscle cells to work together. We don't want some working now and then another second or two, something else happens. No, we want all the muscle cells to contract together. That is accomplished because of the intercalated discs, gap junctions, and desmosomes. So take a careful look at that. We talked a lot about the conduction pathway of the heart. There'll be questions on the conduction pathway. The conduction pathway of the heart is instrumentally important. Take a look at the conduction pathway starting at the SA node. The SA node is the primary node responsible for initiating the conduction of the heart. Know the pathway. SA node, AV node, bundle of His, left and right bundle branches ending in Purkinje fibers. Know the pathway of the conduction system of the heart. If there's a problem in the conduction system of the heart, now we have an arrhythmia. The SA node sets the heart rate. We talked a lot about the fact that if left untouched, the heart rate by the SA node would be too high. It would be over 100 beats per minute. What is the normal heart rate of a human heart? 68 to 72, 73, something like that, right? So what changes it? What slows the heart rate down?
Parasympathetic?
Yeah.
Give me the name of the nerve.
Vagus nerve.
Vagus nerve. Cranial nerve number 10, vagus nerve. Vagus nerve is instrumental because it directly affects the heart rate by its input into the SA node. It would be nice to know the formula for cardiac output. Cardiac output is heart rate times stroke volume. It'd be nice to know that. It would be nice to also to know nodal graphs versus ventricular graphs. Some of you may have messed up that on exam number 1. When the ventricle is contracting, we have a specialized graph to show that. And the important feature on that graph is this plateau, whereby now slow calcium channels are opening and now all of the cells are contracting as a unit. So it would be nice to know what a ventricular graph looks like. Whether it be on the left side, or if we even are contracting the atria, which is a smaller graph, but still it's contracting as a unit. Both atria contract together, both ventricles contract together. So we want to make sure that we get as much blood in the right spot as we possibly can. We went over this in detail. From the cardiac center, which is in what part of the brain? It would be nice to know it's in the medulla. We now are going to be focusing on 2 nerves, the vagus nerve and we also are going to be focusing on the sympathetic nerve system to the heart. So we have parasympathetic and sympathetic distribution to the heart. The parasympathetic supply is the vagus, and that slows the heart rate. The sympathetic supply is coming from the sympathetic chain ganglia, and that actually goes not only to SA node, AV node, but also to cardiac muscle directly, and that increases heart rate. So these 2 components, sympathetic and parasympathetic, are under the division of what major system? What major nervous system? Autonomic nervous system. Autonomic nervous system. It would be nice to know maybe 1 or 2 characteristics of sympathetic versus parasympathetic. Sympathetic has very short preganglionic fibers. Because they synapse in the sympathetic chain ganglia right next door. Then they have very, very long parasympathetic postganglionic fibers. Whereas the parasympathetic supply, coming directly from the brain, cranial nerve number 10, has a very, very long preganglionic fiber, and then it synapses very close to or directly on the organ, and then has a very, very short postganglionic fiber. It'd be nice to know that. It would also be nice to know what is the pharmacological agent of the sympathetic chain division of the autonomic nervous system. What is the pharmacological agent at the end in the target organ here?
Norepinephrine?
Epinephrine or norepinephrine. What is the pharmacological agent of the parasympathetic supply?
Acetylcholine.
Acetylcholine. Be nice to know that. We talked a lot about how the conduction system bleeds into the EKG. So there's going to be a few questions on EKG on the exam. So we're going to have to know what the three waves are, what they represent on the graph paper, and what they represent on the cardiac cycle. So I might say, for example, the P wave on a piece of graph paper represents something, but what does it represent on the cardiac cycle? Okay, so you have to know both of them. You have to know both of them. So the EKG, there will be some questions on EKG. Right? Yeah,
Are
another
you
question.
looking for the name of, in the stage?
If I say if it's on a graph paper, from the EKG machine, from the electrocardiograph machine, right? The P wave represents atrial depolarization. What does depolarization correlate to if I put it on a cardiac cycle? You have two choices: systole or diastole.
Diastole.
No, that was
Diastole.
Yeah.
What does it represent on the cardiac cycle?
No, systole. Polarization systole.
Atrial systole. The atria is contracting. Go over that. What does QRS represent on the graph paper? Ventricular depolarization. As soon as you see the word depolarization, what's happening? It's contracting. What are the names given when we say contraction in the cardiac cycle?
Systole.
Systolic contraction, systole. What is the T wave? T wave is the only one you can see, which is repolarization. So what does that represent? Ventricular repolarization. So on the cardiac cycle, what does that mean? Ventricular diastole. Now the ventricle is filling. So the only thing you don't see on this chart is atrial repolarization. Why?
It's hidden because it's smaller.
Because it's happening right there. It's happening right where the big QRS wave is. And the QRS, graph paper is only 2 dimensions, length and height. You can't see it. It's hidden by the big QRS wave. These isoelectric lines are interesting, these segments. So we have now a wave, we have a segment, we have another segment. Those segments are isoelectric lines. You might want to take a look at that and what that means, right? What's happening in those isoelectric lines. We went over that quite a bit. And we talked a lot about the EKG. We have a lot of varying things concerning arrhythmia. I'm not going to make you memorize all of that for the final, but we know that the atria and ventricles have to beat in a sinus rhythm. So this is important. So the sinus rhythm means it has to beat in a normal pattern, like this. normal sinus rhythm. So when someone says sinus rhythm, that means that we have a normal PQRST complex. When that goes out of sync, we have various things which are determined to be heart block. And so your heart is beating in abnormal rhythm. The king of them all is when the ventricle contracts willy-nilly. They contract crazy. They're not contracting when they're completely filled. That, if worst case scenario, now you have VTAC. Now you have ventricular tachycardia. Right? Now you have to get paddled to return the normal sinus rhythm back to the heart. My friend. Hope I brought that with me, otherwise I have to go get it. Well, I'm gonna talk about this because this will appear on the exam in some manner or form. Let me go back. I have I have that. So you probably want to look at that, right? When I'm talking about it, Cheryl?
Does it matter?
Oh, I have it here.
Okay.
Outsmarting myself is good. I like to do that occasionally. Okay. I didn't think I would forget that. If you don't study anything else for the heart on cardiovascular, study this. Okay, this is my worksheet. This will tell you a lot. And there's going to be a lot of questions that can be asked on the exam regarding this particular worksheet. We know that the two pumps are identical in their operation, whether it be left side or right side, doesn't matter. They're both doing the same thing at the same time but with different structures. But the mechanics is the same. So I've drawn just the left side of the heart. So the left side of the heart is nothing more than the left atrium, the left ventricle, and the aorta. And so I put those three structures. If I were to draw the right, I would have put right atria, right ventricle, and pulmonary trunk. Okay, but both sides are doing exactly the same. There's a lot of stuff going on here. We know that, I like to start on this one first because the cardiac cycle is all of the mechanical events during one systole and one diastole. So what's happening piecemeal? And we have to remember that these I've spread these out dramatically on the piece of paper, but they're happening very rapidly. They're happening very rapidly in the heart, when the heart's working. One of these phases is bleeding into the next very, very quickly, milliseconds apart, right? I've spread them out so that we can understand them a little bit better, but it's happening very quickly. So we have to know that blood through the heart, this is an important concept, I'll put this on there somehow. Blood through the heart flows according to pressure, period. Pressure is the driving force which makes blood move through the heart. As one chamber fills and increases pressure, another chamber is low pressure and is receiving blood. It has to work that way because blood flows in one direction through the heart. So we know that blood is moving from a high pressure gradient to a low pressure gradient, high to low. So as we're moving blood through the heart, we know that we're getting all this blood returning back to the atria, right? All the venous blood is returning through to the atria. Whether it be right or left, all the blood is returning to one of the two atria. And then the atria is getting increasingly involved with increasing pressure. So it's like putting water into a balloon. So pressure is building, building, building. So what is eventually is going to happen is the atria is going to contract and dump its blood into the ventricle. So these squiggly red lines here represent contraction. So the atria at the last bit of blood squeezing, wringing out of the atria, it contracts and squeezes the last percentage of blood from the atria to the ventricle. So now we have blood in the ventricle. We remember now the ventricle was lower pressure than the atria. As blood now starts to flow into the ventricle starts to take over. Now the ventricle has gone from 0 millimeters of mercury, slightly increasing pressure to no more than 5 or 6 millimeters of mercury. That's all it takes. That closes the AV valve. That closes the AV valve, that amount of pressure. That amount of pressure, blood hitting the AV valve, creates a sound. So the first heart sound is S1. So S1 means closing the AV valve. That's the first thing you have to realize on this whole event, right? The blood hitting the AV valve shuts it. Chordae tendinii come into play, they hold it firm, they hold it tight, so no blood is now going to be squirting back into the atrium. The ventricle continues to contract. The ventricle contracts, but it doesn't reach the point where it can spring open the aortic valve until it reaches about 81 millimeters of mercury. Remember, when blood is flowing into from the atria and the atria is contracting, this semilunar valve is shut. That semilunar valve is shut. The only valve that's open is the AV valve. So when the ventricle starts to contract, it shuts the AV valve, but we already we have closed previously the semilunar valve. When the pressure in the ventricle reaches a certain millimeter of mercury, now it pushes open the semilunar valve of the aorta. So now we go from a situation where both valves are shut. All valves are closed. Say that one more time. All valves are closed. That's a situation called isovolumetric contraction, where you hear S1. Quickly, when the pressure reaches a certain degree, it opens the semilunar valve. AV valve is still shut. Now we have a rapid ventricular contraction phase where now blood is rushing into the aorta. The pressure which is generated in the ventricle at its peak is about 120 millimeters of mercury. So it rises from 81, it keeps contracting, keeps ringing, and so now we get up to about 120 millimeters of mercury pressure-wise, and that pressure is transmitted directly to the aorta. The pressure from the ventricle is transmitted to the aorta. The aorta swells in size to accommodate the increased amount of blood. So we have blood now rushing through the semilunar valve into the aorta. That quickly moves to a slow ejection phase where now the pressure in the ventricle starts to decline, because most of the blood is now pushed into the aorta. So the pressure in the ventricle starts to drop. So the pressure starts to be reduced from 120 millimeters of mercury downward. This is the last part of the ejection phase. It's called the slow ejection phase. I could have just put these two phases together, rapid and slow. They are very, very closely aligned. I've just spread them out so that you can really see what's going on a little bit. The pressure in the aorta now is still higher than the pressure in the ventricle. So this quickly moves to this phase right here. The ventricle, after complete ejection, there's some blood that's always left over in the ventricle. That is called end systolic volume. You should know that worksheet where I have listed all those initials: ESV, EDV, CO, HR, SV, all of those initials. You should know that worksheet very carefully. There are going to be questions on that from that worksheet. So now we have some blood that's always left over after the ventricle has done its work. There's always about 50 milliliters of blood left over, which is end systolic volume. It's after systole has been completed, there's always blood remaining. But the pressure now in the aorta is higher than the ventricle. Because the ventricle is now reducing in pressure, the pressure is still high in the aorta. The pressure in the aorta pushes now backward and closes the semilunar valve. When the blood hits the semilunar valve and closes the semilunar valve, we hear another sound. That's S2. S2 is related to isovolumetric relaxation phase. Isovolumetric relaxation phase. Now the ventricle is going to begin filling with blood. So we have two phases that we hear a sound: isovolumetric contraction, isovolumetric relaxation. S1, S2. You should get those in your mind. As soon as you hear the word isovolumetric, that means that no blood is coming in and no blood is going out. So all valves are shut for that split millisecond in time. Once we know now from the relaxing phase, now we have blood continually entering the atrium. All along here, blood is coming into the atria. And now what happens is when the atria starts to get high in its pressure, it forces open the AV valve. So now blood is rapidly filling, coming through the AV valve into the ventricle again with no resistance, just a massive flow of blood back into the ventricle. This quickly goes to the slow phase where now we have more filling coming in from the atria, and eventually what we are going to end up with through the rapid and slow phase of filling, we're going to end up with about 80% of the ventricle filled. So we have ESV, and then we have the rapid phase, and then we have the slow phase, and that equals about 80% of the ventricle being filled. And then what happens very, very quickly, we go to the last 20%, where the atria now starts to contract and squeeze out the last remaining part of blood that it has in it to be put into the ventricle. And usually that's about 20%. So now we have the ventricle filled again, and now we start the cycle again. Right? We repeat this cycle again. And so that's one systolic and one diastolic event in the cardiac cycle. Yes?
What does S3 and S4 signify here?
S3 and S4?
Yeah.
You want to get clinical? Or do you want to just stick with your final? S4 is a clinical event. You can hear another heart sound. If you're very, very trained cardiologist, can hear another heart sound, which is S4. S4 is related to this phase here where the ventricle does not fill nicely with blood because it is stenotic. The ventricle is somehow too inelastic to expand. That's a simple way to say it. So the ventricle is stenotic, there's calcified cells in the ventricle doesn't have enough elasticity to absorb all the blood from the atria. So that creates a sound. That's a pathologic sound. You can hear S4. S3 is just after complete exercise. If you're out there running around violently, running around playing soccer, running around doing exercise, vigorous exercise, there's another sound that you can hear through vigorous exercise with the rapid filling phase, and that is S3. So there's a couple other sounds you can hear clinically, but you guys don't have to worry about that. I want you guys just to focus on S1 and S2, where they occur on the cardiac cycle, and what's going on in S1 and S2, right? This chart over here is the chart that is in your book, and this chart is a very, very nice chart because it supplies all of the data that we just talked about over there. So we know, for example, if we just look at the volume of blood, we know that when the atria has a large amount of blood, it is related to EDV, end diastolic volume. That's getting the most amount of blood back to the heart, right? And we talked about various pumps that accomplish that, getting blood back to the heart. The muscular pump is probably the most important pump. Remember that? It squeezes blood in the veins and pushes blood toward the heart. We want to get as much blood back to the atria as we possibly can, end diastolic volume, because that is the only amount of blood we're going to be able to use to put into the ventricle. Whatever we have back in the atria is going to be the blood we can put to the ventricle. We increase the amount of blood that we can put back to the atria, and then the ventricle is going to contract. The ventricle is undergoing contraction, so now it's going to be reduced, the amount of blood left in the ventricle, and that's going to be ESV, end systolic volume. The amount of blood that we normally can push back through venous return, remember, venous return is the key. Venous return is the key of getting blood back to the heart. It has to come back through veins. So venous return is essential for this whole operation to work. You'll probably see that somewhere. Veins are the most important factor. It usually is about 120 milliliters of blood. ESV, after the ventricle contracts, it usually leaves about 50 milliliters in it. The difference between those two volumes is the amount of blood that's ejected. That is stroke volume. So 120 minus 50 is what? 70. So that's the amount of blood really that is concerned with stroke volume. So we can look at cardiac output, and we know now, oh, I have wrong pants, slacks on today. I don't have slacks on. We know cardiac output is heart rate times stroke volume. And we know that that's in milliliters. But I might get tricky and ask for liters. How many milliliters in a liter?
1,000.
How many?
1,000.
1,000. If we look at this one, this line is sort of an interesting line. When we look at the pressure, we can look at the pressure being going up to the fullest extent, about 120 milliliters of pressure. when the ventricle contracts. And we see a thing called the dicrotic notch. The dicrotic notch is sort of an interesting event because when now this valve is shut, the semilunar valve is shut in the relaxation phase, the aorta has higher pressure than the ventricle. So the blood starts to rush back to close that valve. When the blood rushes back to close the valve, we hear a blip, and the blip is on the chart as the dicrotic notch. It sort of ricochets and bounces off the valve. Boom. That creates that blip called the dicrotic notch. Okay. We know that the EKG is important because we know the electrical event has to happen slightly before the heart mechanical event. So we have to have electrical current going to the atria, going to the ventricles, slightly before they can contract. It happens very, very quickly, but slightly before the ventricle actually can mechanically contract. It has to be stimulated electrically. Right? So we see the EKG graph slightly ahead of where the atria contracts and where the ventricle contracts. Any question on that? I would really recommend that you know this worksheet. Know this worksheet. Right? And know the mechanism involved with isovolumetric phases. We talked a lot about where you can hear the heart. Might be nice to know where you can hear the heart when you put a stethoscope on a chest wall. Where you hear the mitral valve, where you hear the tricuspid valve. The pulmonary and semilunar aortic valves are opposite. Most people think the aorta is on the left, so the aortic valve is over there. No, the aortic pulmonary and the aortic semilunar valve is on the right. Pulmonary is on the left, so it might be nice to know where you can hear them. We talked a lot about problems in heart defects. So I might throw one clinical question in there somewhere. It would be worded in a way that you would understand it, like what would happen if you wanted to mix blood between the two atrias? What would have to happen if there was mixing of blood between the two atrias? You'd have to have some kind of hole in the heart between the two atrias. When in fetal life, there is a hole there. What is that hole called? What is that opening called? I think you should look at that and make sure you know that opening. Because by the time it's an adult, it has to be closed. So you should know the names of both of those. Because we don't want mixing of blood. We know the right side is deoxygenated, the left is oxygenated. We don't want the two to mix. Okay, I'm going to stop there on cardiovascular. Anybody have some questions that if you've been looking at your notes or whatever that have come up that bother you relating to cardiovascular issues? We talked a lot about the importance of venous return, how blood has to get back to the heart, because we need as much blood back to the heart as we can possibly get in order to pump it out. So venous return is essential in this whole thing. Remember the laws of the heart, Frank Starling law of the heart, venous blood, it preloads, right? There's preloading and afterloading. Preloading is getting as much blood back to the heart as we possibly can. Afterload is concerned with the ventricle contracting and pushing blood out. So we have preload and afterload. That's on that worksheet with all the other initials on it. ESV, EDV, CO, and all that. You should know, you should look at that worksheet. That's a very, very important worksheet. Did you have a question?
Does cranial nerve 10 only innervate the SA node, and then the rest of like the conduction pathway kind of takes over from the SA node, or does the cranial nerve 10 kind of innervate everything in the heart, in the conduction pathway?
On the parasympathetic side?
Yeah.
The parasympathetic side, no. The parasympathetic hits the SA node and the AV node and controls the rhythm of those two nodal focuses. The sympathetic node hits the SA node and the AV node, as well as the heart muscle itself in the ventricle. But you should know the normal conduction pattern pathway through the heart, from SA node to AV node, bundled is left and right bundled branches or Purkinje fibers. And the complication from that is arrhythmia, right? The complication is that is arrhythmia. Make sure you, again, make sure you look at the EKG and being able to correlate the EKG waves to what's happening in the cardiac cycle. Okay. Any other questions that have popped up along your studying situation? You can think about any other questions you might have. I'm going to give another review session. I'm going to talk about respiration and urinary the next time. And I might even have throw in there a little bit about acid base, because those two systems are not as complicated as the heart. The cardiovascular system is going to occupy a significant amount on this exam.
How heavy did you say the digestive system was? I didn't hear it earlier.
The digestive system is I will mention the digestive system next Friday, but you should be looking at that now, and you should be going over the worksheet where I put the digestive enzymes and the digestive hormones. The digestive enzyme that a lot of people sort of missed up on the last exam was CCK, cholecystokinin. CCK is really an important hormone. Really an important hormone, as is secretin. CCK and secretin are both major hormones produced in the duodenum, and the importance of secretin versus cholecystokinin is that in secretin it contains bicarbonate. It contains basic bicarbonate ions. That's really important because you've got to remember the food entering the small intestine is coming directly from the stomach. And the stomach is full of acid. So the food from the stomach, called what?
Chyme.
Chyme, is acidic. And you have to be able to somehow neutralize the acid for the digestive enzymes to work in the small intestine. How do we do that? By the direction of the hormone secretin. Secretin also does the same things as CCK. It increases pancreatic enzymes, decreases gastric secretions, but it also adds bicarbonate. It adds bicarbonate ions which neutralize the acid from the stomach. So those two hormones are really important, CCK and secretin. Gastrin is a hormone that causes gastric secretions and the stomach to increase its contractibility, to churn up the food. So the hormones are very, very important on that worksheet, as are the digestive enzymes. And basically, I think everyone did fairly well on the exam, but know exactly what enzyme, the specificity of enzymes. has to work on a certain substance, has to have the right temperature and the right pH, otherwise it doesn't work. And only enzymes digest food. Does hydrochloric acid digest anything?
No.
Nope. That was a question on the exam.